What if I told you that the exact same biological glitch that causes, like, really terrifying brain diseases?
Yeah, it's molded proteins, right?
Exactly,
Yeah.
misfolded protein. What if I said that glitch is actually the missing blueprint for building a galaxy-spanning technological empire?
Yeah.
Welcome to the deep dive.
Well, it is a premise that fundamentally rewrites how we think about material science
Right.
and energy generation, and honestly, the very definition of what a civilization even is.
Yeah, totally.
Because when you first encounter the idea, I mean, it sounds completely untethered from reality.
Completely untethered. And you hear misfolded protein,
Yeah.
and you immediately think of biological chaos, right?
Yes, absolutely.
Like a single protein folds wrong, and then it just forces everything around it to fold wrong, too, in this, unstoppable chain reaction.
Pathogenic cascade.
Right. It's the mechanism behind some of the most devastating neurodegenerative diseases in human history.
Absolutely unequivocally clear. We are not talking about infectious disease-causing mammalian prions here.
Right.
We are not talking about mad cow disease or
No.
anything that causes pathology in humans or animals.
Yeah, nobody is suggesting we build spaceships out of a literal disease.
Oh,
that would
no.
be like the most terrible sci-fi movie ever.
Precisely. So, for this deep dive, we are unpacking a massive, highly technical 2026 manifesto.
A huge document.
Right. And its associated addendums written by an independent researcher named Zachary David Enfield.
Yeah.
The work is titled Prion and Amyloid Architectures, a total application framework.
Mm-hmm.
And the framework explicitly relies on yeast prion determinant, specifically looking at proteins like sub-35,
Yeah.
as well as entirely synthetic lab-created amino acid sequences.
So we are stripping away the pathology entirely.
Completely. We are just borrowing the architectural logic of self-assembly. We are taking the microscopic mechanism that allows a protein to, template its own precise geometric shape onto other ambient proteins.
Right, just the mechanism itself.
It is exactly like how modern medicine uses the self-replicating machinery of a retrovirus to deliver targeted gene therapy.
Oh, sure.
You do that without giving the patient the actual virus. We want the delivery
We
mechanism.
want the amplifier.
Exactly. We do not want the disease.
Okay, with that massive disclaimer out of the way, let's look at the absolute absurdity of what this framework proposes.
It is quite a leap.
It really is. We are going to explore how a single microscopic biological structure could theoretically be used to, like, print synthetic food,
Yes.
build solid-state nuclear batteries,
Yeah.
power thermodynamic quantum computers, and ultimately construct Dyson swarms around distant stars.
Which sounds like magic.
It totally does.
Yeah.
And we are going to trace this entire journey logically.
Right.
We'll start by looking at what this material actually is and why it's so indestructible.
Because you have to start with a physical substrate.
Exactly. Then we have to figure out how to shape it.
Yeah.
Because, a pile of indestructible sludge isn't a machine.
Far from it.
After that, we'll look at how to power it, how to make it compute, and finally, what happens when you launch this whole crazy technology into the vacuum of space.
And while the synthesis of all these ideas is radical, the most crucial point to keep in mind is that every individual component we are going to discuss today is grounded in peer-reviewed experimental literature.
They are real.
These aren't just thought experiments. These are physical realities sitting in laboratories right now.
Wow. Okay, so let's start at the very bottom. The physical foundation.
Let's do it.
The core superpower we are talking about here is something called the cross-beta architecture.
Yes.
Enfield's manifesto explains that all amyloid and prion-derived fibrils share this very specific structural motif.
Right, the cross-beta.
Right. So when these proteins misfold and snap together, they form these beta strands, and these strands orient themselves perpendicular to the long axis of the growing fiber.
Like rungs on a ladder.
Exactly, like rungs on a ladder, while being hydrogen bonded into incredibly dense sheets that run parallel to the axis.
If we connect this to the broader picture of structural engineering, what nature has managed to do here at the nanometer scale is create a true nanocomposite material.
Okay, composite.
Think about how we build modern armor or aerospace components. We don't just use a single solid block of metal.
Right, that would be too heavy or brittle.
Exactly. We use composites. We layer rigid materials with flexible material.
Oh, I see.
In the cross-beta architecture, you have alternating layers of very rigid, densely hydrogen-bonded beta sheets.
Okay.
And they are separated by the amino acid side chains that basically act as shock absorbers.
So it's not just a rigid crystal that shatters when you hit it.
Exactly. It's the same design logic you see in, like, an abalone shell.
Oh, the inside of an oyster shell.
Right, which is made of a material called nacre.
Yeah.
Or, even in your own human tendons.
Oh, wow.
You are combining extreme stiffness with massive energy dissipation.
Mm-hmm.
But here, it is operating at a scale of just 10 nanometers wide.
And the performance numbers on this are just wild.
They really are.
Let's look at the data from the sources. Engineered polymeric amyloid reaches a toughness of 160 megajoules per cubic meter.
Which is an astronomical figure.
Right. Now, to put that in context for you listening, natural spider dragline silk has always been held up as, like, the holy grail of biological materials. Decades, scientists have been trying to figure out how to harvest spider silk to make bulletproof vests and, elevator cables because it is so incredibly tough.
Yes.
Spider silk comes in around 150 megajoules per cubic meter.
So this synthetic amyloid framework actually beats the holy grail of biomaterials.
It beats spider silk. And it does this at 1/8 the density of high tensile steel.
It's important to be precise about what toughness means here.
Right, clear that up.
Because people often confuse it with tensile strength.
Oh.
It doesn't match steel in ultimate tensile strength.
Mm-hmm.
Meaning, a thick cable of steel can take more raw pulling force before it just snaps in half.
Right, so you couldn't tow a truck with a single thin strand of it compared to steel.
Exactly. But toughness is the measure of the total amount of energy a material can absorb before catastrophic failure.
Oh, I see.
It's about how much the material can deform, stretch, and distribute an impact without breaking.
Okay, that makes sense.
In terms of toughness, the amyloid fiber is vastly superior to steel.
Wow.
And it is doing this while being completely biocompatible and capable of replicating itself in a liquid solution.
But, the chemical stability is what really threw me.
Oh, it's fascinating.
Because when I think of biological materials, I think of things that are really fragile.
Sure.
Like, if you heat up a protein, it denatures. Think about cooking an egg. The clear part turns white because the proteins are, like, unfolding and tangling up due to the heat.
Denaturation, exactly.
Right. Biology is usually a delicate flower. But according to the data Enfield aggregates, fibrils formed from these specific synthetic sequences are functionally indestructible.
It's true.
They survive being soaked in pure formic acid.
Mm.
They completely resist protease degradation, meaning the enzymes that normally chew up proteins in your body can't even scratch them.
The radiation hardening is perhaps the most significant property for its future applications, though.
Oh, man, yeah. Let's talk about that.
These fibers can survive doses of gamma-ray radiation that would absolutely destroy standard silicon logic boards.
Let's expand on that because that is huge. Why does silicon fail in space and why does this survive?
Well, think about a traditional computer chip made of silicon. It works by moving tiny electrical charges around to represent ones and zeros.
Right.
When a high-energy particle from space, like a gamma ray, smashes into that silicon,
Yeah.
it acts like a bowling ball hitting pins.
Oh.
It knocks electrons loose, creating an ionizing path. This uncontrolled electrical charge can easily flip a 0 to a 1, corrupting data.
A literal bit flip.
Yes. Or it could physically burn out the microscopic transistors.
Yikes.
This is exactly why satellites and spacecraft require massive, heavy lead shielding or incredibly expensive, redundant, radiation-hardened chips.
Which adds so much weight to the rocket.
Exactly. But the cross-beta amyloid structure doesn't rely on those fragile crystalline electron paths in the same way.
How does it handle the hit, then?
The molecular bonds are so densely packed and the structural redundancy is so high that even if a gamma ray shears straight through a section of the fiber,
Yeah.
the surrounding hydrogen bonds hold the overall geometry intact.
It just holds its shape,
It physically cannot fall apart.
which makes it, like, the ultimate disruptive material for aerospace or deep-sea exploration or high-energy electromagnetic environments.
Absolutely.
But the real genius of the cross-beta superpower, according to the manifesto, is something called hydrogen bond density, or HBD. Enfield refers to this as the engineering dial.
The HBD is the master control variable.
Okay.
The mechanical and electrical properties of these fibers are directly proportional to the density of the hydrogen bonds in the core of the fibril.
So if I'm understanding this dial correctly, it's all about how tightly we pack these microscopic connections.
Exactly.
Like, if I want a material that acts like a bridge cable, I pack them incredibly tight.
Right.
But if I want, say, a temporary medical scaffold for a broken bone that will eventually melt away.
You loosen them up.
I loosen them up. Is it really as simple as just swapping out a few lines in the genetic code to turn this dial?
It is exactly that elegant, yes.
Wow.
The entire structure is dictated by the sequence of amino acids.
Okay.
If you want a stiffer, stronger, fibril something approaching a structural steel analog,
Yeah.
you tweak the genetic sequence to use amino acids that naturally pack together incredibly tightly,
Leaving no gaps.
leaving zero microscopic gaps. This maximizes the hydrogen bond density. The layers pull together like a vise. On the other hand, if you want a more dynamic, fragile fibril, perhaps one that easily fragments so that those fragments can float away and seed new growth elsewhere.
You decrease the density.
You decrease the hydrogen bond density. You do this by introducing disordered amino acids or polar-rich cores that repel each other slightly.
That is amazing.
You are literally tuning the physical-mechanical spectrum of the material from a liquid-like gel all the way to shatterproof glass.
Just by altering the alphabet of the amino acid sequence before you even grow it.
Precisely.
And that sequence dictates the electrical properties too, right?
Yes, it does.
The sources mention a 2021 study published in the Proceedings of the National Academy of Sciences by Ships and colleagues.
A landmark paper.
They demonstrated that these biological fibers can conduct electricity intrinsically.
Right.
Like, you don't have to coat them in gold or copper. They do it naturally through a process they call tyrosine hole hopping.
Tyrosine hole hopping is a fascinating quantum mechanical phenomenon.
It sounds like a dance move.
It really does. But tyrosine is an amino acid that has a prominent aromatic ring.
Okay.
It's a little hexagonal structure of carbon atoms.
So think of those tyrosine amino acids like a line of microscopic stepping stones placed across a river.
Well, that's a good way to look at it.
Because of their unique ring-like chemical structure, an electron doesn't have to literally swing through the water of the cell.
No, it doesn't.
It can literally hop from one stone to the next across micrometer distances.
That is a perfect visualization. When the cross-beta fiber folds, the geometry forces these tyrosine rings to stack up perfectly.
One on top of the other.
Exactly, one on top of the other, inside the core of the amyloid fiber.
Wow.
It creates a continuous, unbroken pathway. It acts as an organic semiconductor.
That's wild.
But it gets even more versatile. Other variations of these fibers, like those studied by Navarro in 2024,
Oh, I read about that one.
they don't just conduct electrons. They act as incredibly efficient proton wires.
Photon wires.
Yes. They pass positively charged hydrogen ions down the line faster and more efficiently than the synthetic biomaterials we currently use for things like hydrogen fuel cell membranes.
So if we pull all of this together, imagine a microscopic roll of Kevlar.
Okay.
It's incredibly tough, it's radiation-proof, it can conduct electricity, and it can act as a semiconductor.
Yes.
But here is the massive kicker. Instead of needing a billion-dollar industrial chemical plant that requires massive heat and pressure to manufacture this Kevlar.
Right, like traditional manufacturing.
Exactly. It can literally grow and replicate itself in a bucket of water at room temperature.
Yes, it can.
You just drop a single microscopic seed fragment into a vat of amino acids, and the chain reaction begins. It builds itself.
That is the self-assembling manufacturing logic that makes this framework so potent.
It's almost free.
The energy cost of production is practically zero compared to traditional industrial manufacturing.
But okay, let's unpack this. Here is where I have to step in and, push back on the premise a little bit.
Please do.
If this material is this tough and this chemically stable and it just grows whenever it touches free amino acids,
Yes.
isn't it just an inert brick?
Ah,
I mean, a very impressive, tiny, indestructible brick, but still a brick, or rather like a vat of super strong conductive sludge.
I see where you're going.
How does it actually do anything active? How do you make a specific shape out of it or build a machine rather than just filling a swimming pool with super Kevlar?
That is the essential hurdle.
Yep.
And it moves us precisely to the next critical problem of the framework.
Okay.
It's entirely correct. A seeding cascade, once initiated, just propagates wherever monomers are available.
Monomers being the free-floating building blocks.
Yes, it grows in all directions simultaneously.
Just a big blob.
It fills a volume, but it doesn't write a pattern. It is an incredible canvas, but a canvas is entirely useless without an architect to draw the blueprints.
Exactly.
We need spatial control. We need a way to tell the protein exactly where to grow, what shape to take, and when to stop growing.
Okay, so we have this indestructible protein sludge. But a vat of sludge isn't a microchip.
No.
How do we actually force these chaotic proteins to snap together into a specific, intricate shape without physically touching them?
This is where we get into prion optogenetic lithography.
Right. The manifesto calls this solution prion optogenetic lithography. And this is where Enfield takes three entirely different pillars of modern science and just smashes them together.
He does. Pillar 1 is the prion self-assembly we just spent time dissecting.
Right, the amplifier.
That is the engine that does the building. Pillar 2 is optogenetics.
Optogenetics.
Optogenetics is a field of biology where we use genetic engineering to encode light-responsive protein domains into cells.
Light responsive?
Yes. We literally steal the mechanism that plants use to turn their leaves toward the sun.
Oh, wow.
And we insert it into other proteins. These are domains like Cry2 or LOV2 that physically change their 3D shape in response to very specific wavelengths of light.
Okay, so that serves as our spatial gate.
Exactly. And pillar 3 is photolithography, which is the foundational technology of the entire semiconductor industry.
Which is how we make computer chips today.
Right. It's the process of using highly structured microscopic light to write intricate circuit patterns onto silicon wafers at nanometer resolution.
So we're combining the self-growing nature of the protein with a light-activated switch and guiding it with a laser.
Yes.
To make this work, the manifesto points to something called the phobit integration, which was a real concept published in Nature Communications in 2025. It's basically a modular tag.
This is where the molecular engineering becomes truly elegant.
Okay, break it down.
You engineer a chimeric protein, a Frankenstein molecule, essentially.
Nice.
You physically attach your light-responsive domain, your LOV2, to your prion-forming domain, your SUB35.
You glue them together.
Exactly. Now, in the dark, the light-responsive domain acts exactly like a mechanical lock.
Oh, I see.
It folds over the prion domain and physically restrains it. It holds the monomer in a harmless, non-assembling state.
So it can't grow.
It cannot fold into the amyloid structure. It just floats there, inert.
But the moment you strike it with a specific wavelength of light,
Say, a highly precise blue laser.
yeah,
The photon hits the LOV2 domain. The energy from the light causes the chemical bonds in the domain to shift.
the lock disengages.
It swings open, the prion monomer is unleashed, it is finally allowed to adopt its assembly-competent shape.
And it snaps into place?
It instantly snaps into the growing fiber. But it only happens in the exact microscopic location where the light touched it.
So the light is literally the printer.
Yes.
We have to completely change how we think about 3D printing. Like, you don't have a nozzle squirting out hot plastic or liquid metal layer by layer.
Got it all.
You have a giant vat of this clear protein soup, and you shine a complex three-dimensional holographic light pattern deep into the center of the vat. And the fiber only grows where the light exists.
If we connect this to the bigger picture of human manufacturing history,
Okay.
we are moving from a mold paradigm to a light paradigm.
That's profound.
For thousands of years, to make a complex shape, we created a negative space, a mold out of clay or iron, and poured molten material into it.
Right, like casting a sword.
Or we used subtractive manufacturing, taking a block of wood or stone and carving away what we didn't want.
Like a sculpture.
Exactly.
Yeah.
Even modern cutting-edge 3D printers still just extrude materials sequentially.
Yeah, layer by boring layer.
But here, you use light to command a fluid to crystallize into a highly specific three-dimensional structure from within.
Wow.
The physical printer is nothing more than a light source. The complexity is entirely virtual. It exists in the software that shapes the light field.
Think of what you could do with this. The sources talk about in vivo tissue engineering.
A prime example.
Imagine you're a patient who has suffered a massive traumatic wound or a severed spinal nerve.
Devastating injuries.
Currently, doctors might try to surgically implant a rigid plastic scaffold or take bone grafts from your hip.
Highly invasive.
Very. But with this, you don't do massive surgery. The doctor injects this harmless, dark-state optogenetic protein soup directly into the wound cavity.
Right.
Then they use a device to shine a precisely patterned medical-grade laser through your skin directly into the injury. The scaffold literally grows itself along the light paths, perfectly matching the jagged, complex geometry of your specific wound.
It provides an instant custom-fit lattice for your own natural cells to grab onto and regenerate along.
And tying it back to what we discussed earlier, because you control the hydrogen bond density via the genetic sequence.
The dial.
The dial. You can design that exact scaffold to slowly dissolve over a period of exactly 6 weeks.
Melting away just as your natural tissue permanently replaces it.
That is just incredible. They also talk about food, which sounds crazy, but protein lattice printing could change everything.
It addresses a massive logistical problem.
The goal here is creating the exact microarchitecture of meat. Think about a premium steak. The reason it has that specific chew isn't just the protein itself. It's the bundles of aligned protein fibers, the specific texture.
The mouthfeel.
Right. Right now, lab-grown meat is mostly just mush. But with optogenetic lithography, you don't need a cow.
No.
You just grow the perfect microscopic protein lattice with light, layer by layer, aligning the fibers perfectly. Infield points out this is the holy grail for NASA.
Indeed, mass is the enemy of space travel.
You can't bring a herd of cows to Mars.
You don't even pack tons of pre-made, freeze-dried food for a 3-year Mars mission.
Really?
You pack a highly compact, dense cartridge of raw amino acid soup, and you print the structural lattice of a steak on demand, customized for the nutritional needs of the astronaut that day.
That is wild. And we can't forget the electrical applications either.
Yeah, we cannot.
Because we know these fibers can be tuned to be conductive through that tyrosine hole hopping we talked about. We can use light to print highly complex three-dimensional wiring networks.
It's suspended entirely in gel.
You can grow nanocircuits completely suspended in a block of hydrogel. You can't do that easily with traditional silicon chip fabrication.
Silicon is inherently flat.
Right. It's layer-by-layer lithography built on a 2D wafer. This allows for truly 3D interwoven circuitry.
It fundamentally removes the geometric constraints of modern electronics.
It was literally like a sci-fi replicator, but instead of the ship's computer assembling atoms out of thin air using magic energy fields, it's a laser pointer guiding an army of microscopic biological builders.
That's a very apt analogy.
But I have to ask,
Yes.
what happens when the light turns off? I mean, once that cascade starts, once the protein folds and recruits the next one and the next one, does the fiber just keep growing forever out into the dark?
This raises a profoundly important question.
Like, if I shine a flashlight into the vat, do we end up in a sci-fi gray goo scenario where this tough, indestructible fiber eats all the protein in the vat and bursts out of the container?
It's the biological containment problem. And the framework explicitly addresses this, because without containment, it is a weapon, not a tool.
Right.
The length of the cascade, how far the fiber grows past the beam of light, is governed by two main factors:
Okay, what are they?
monomer concentration and temporal light control.
Let's start with monomer concentration.
First, the chemical reality. The cascade can only grow as long as there is free, light-activated monomer immediately available at the growing tip.
Okay.
If you heavily restrict the overall concentration of the soup, the fiber starves and naturally terminates the moment it leaves the high-concentration light zone.
Oh, so it runs out of bricks to build with.
Exactly. Second, the Fobat system can be designed to be strictly reversible.
Reversible how?
When the light turns off, the LOV2 domains don't just stay open, they naturally revert to their locked state.
The lock snaps shut.
Yes. Any monomer that hasn't successfully attached to the fiber suddenly folds back up and caps itself, preventing further growth.
That's a great failsafe.
And for extreme safety, especially in uncontrolled environments, the framework introduces what are called genetic dominant negative inhibitors.
Okay, dominant negative inhibitors. That's a mouthful. Let's translate that for the audience.
Studies.
Think of these like molecular caps or plugs. They are engineered proteins that are specifically designed to bind to the growing end of the fiber, but they have no sticky side of their own. So once they attach, the chain reaction hits a permanent brick wall.
Yes. You flood the solution with a low concentration of these caps.
Okay.
As long as the light is intensely shining, the regular building blocks outcompete the caps.
They build faster than the caps can plug it.
Exactly. But the millisecond, the light dims.
A cap snaps on.
A cap snaps onto the end of the fiber and permanently halts the cascade. It provides a highly localized, highly controlled reaction volume.
Wow.
The light defines the boundary of the blueprint, and the chemical kinetics provide an absolute stopping point.
Okay, so we've solved the structural problem. We can print indestructible circuits and biological machines with light, and we can stop them from turning into gray goo.
We have the chassis.
But if you build an incredibly complex 3D biological nanocircuit, it is going to need an equally revolutionary power source to run it.
Power is always the bottleneck.
Right. You can't just plug a traditional lithium-ion battery into a microscopic strand of protein. A AA battery isn't going to cut it.
No.
Which brings us to the energy solution. The prion nuclear convergence and piezoelectricity.
This section of the manifesto is perhaps the most paradigm-shifting synthesis in the entire document.
It's nuts.
Enfield is combining advanced nuclear decay physics with biological self-assembly.
It starts by examining a 2024 paper published in Nature by researchers at Soochow University.
Yes.
They introduced something called the coalescent transducer principle.
The brilliant concept.
And when I read this, it completely blew my mind because it solves a problem that has plagued nuclear batteries for decades.
The gap problem.
Exactly. Let's explain how a normal nuclear battery, a beta-voltaic cell, works. Usually, you have a lump of radioactive material, let's say an isotope like nickel-63.
Okay.
You put that radioactive lump next to a semiconductor panel. As the nickel naturally decays over decades, it shoots out high-energy electrons called beta particles.
Like tiny bullets.
Right, like tiny bullets. Those particles travel across a physical gap, smash into the semiconductor, and knock loose electrons to create an electrical current.
It is the conventional architecture, yes. It's the same basic concept that powered pacemakers in the 1970s before lithium batteries were perfected.
Wow, pacemakers.
Yes, and it has a foundational flaw in physics known as self-absorption.
Self-absorption.
As those beta particles travel through the dense radioactive material itself, and then across the microscopic gap to the panel, they constantly collide with other atoms.
Right, they hit traffic.
Every collision strips away massive amounts of kinetic energy, turning it into useless heat before the particle ever reaches the semiconductor transducer.
So it loses power before it even gets to the panel.
As a result, current beta voltaics have a terrible energy conversion efficiency rate, usually hovering in the single digits. Most of the nuclear energy is completely wasted.
But the Soochow University researchers figured out how to solve this. They realized the gap is the enemy.
Yes.
So they coupled the radioisotope directly to the transducer at the molecular level.
They merged them.
They put the decaying radioactive atom inside the crystal lattice of the material that is catching the energy. And by eliminating that travel gap, they achieved an 8,000-fold efficiency enhancement.
8,000 times more efficient. It is a staggering leap.
It's unheard of.
It transforms the entire concept of the beta voltaic cell from a niche, ultra-low-powered device used only in deep space probes into a highly viable long-term power source.
But there was a catch to the Soochow experiment.
There was.
They used a highly synthesized lanthanide coordination polymer.
A very nasty chemical.
Right. It's incredibly complex, toxic, and tremendously expensive to manufacture at scale.
And this is where the biological twist comes in, and where Enfield's framework gets terrifyingly brilliant.
Terrifying is the right word.
He proposes using CRISPR gene editing to insert specific uranyl-binding motifs directly into the genetic code of the amyloid scaffold.
Okay, let's pause. Uranyl binding motifs.
Yes. Now, this isn't science fiction. There are already engineered proteins in existence today, like the superuranyl binding protein detailed in a 2014 Nature Chemistry paper.
Okay.
They can bind uranium atoms out of ordinary seawater with ridiculous precision.
Seawater.
Yes, we're talking femtomolar affinity. They ignore the salt, they ignore the calcium, they only grab the uranium.
That's crazy.
So you engineer your amyloid fiber to naturally bind uranium atoms at precise, repeating intervals along its entire structure.
So they just snatch the uranium right out of the water as they grow.
Exactly. What you achieve is the exact coalescent transducer geometry that Soochow University discovered, but assembled entirely by cheap biology.
Wow.
The uranium atom sits perfectly suspended inside the piezoelectric conductive protein scaffold.
No gap.
Zero gap. When that uranium atom decays, it doesn't shoot a beta particle across a gap. Every single particle, every recoil vibration of the nucleus, every microscopic phonon couples instantly and directly into the conversion medium surrounding it.
Because it's literally inside the medium.
Yes.
And because of the unique properties of the cross-beta structure we talked about earlier, it harvests that nuclear energy in 3 completely different ways simultaneously.
Trimodal harvesting.
Trimodal harvesting. First, it acts exactly like a beta voltaic cell, catching the emitted beta particles to create current. Second, it harvests the thermal gradient. It uses the temperature difference between the hot decaying inside of the fiber and the cooler liquid outside to generate power.
Like a tiny steam engine.
Right. And third, it acts as a piezoelectric generator.
The piezoelectric aspect cannot be overstated here.
Explain piezoelectricity for us.
Piezoelectricity is a phenomenon where certain materials generate an electric charge under physical mechanical stress. If you squeeze or bend a piezoelectric crystal, it produces voltage. This is how the clicker on a gas grill works.
Oh, when you push the button and it goes click.
Exactly.
Yeah.
A tiny hammer hits a quartz crystal inside, generating a spark.
I never knew that.
When a nuclear decay event occurs inside the protein lattice, it is violent. It physically jolts the lattice.
Like a microscopic explosion.
It creates a massive mechanical vibration, a shock wave called a phonon.
Right.
The cross-beta amyloid structure is inherently piezoelectric. As the shock wave ripples through the alternating rigid and flexible layers, the material flexes.
And squeezes the electrons.
Yes, it squeezes electrons down the conductive tyrosine pathways. It literally turns the mechanical shock wave of the nuclear explosion into usable electricity.
That is just wild.
Conventional nuclear batteries just lose all that mechanical shock as wasted heat. This architecture catches it.
It's literally catching the bullet, the heat from the gun barrel, and the physical recoil of the shot, all at exactly the same time.
A very violent but accurate metaphor.
And the crazy thing is, you can use that piezoelectric property even without the radioactive uranium.
Yes, you can.
The sources say that these amyloid fibrils can generate up to 1 volt of electricity just from being exposed to simple acoustic ultrasound.
1 volt from sound waves.
Think about what you could do with that. You could inject these harmless fibers into a solid cancer tumor.
Okay.
Then a doctor waves the standard external ultrasound wand over the patient's skin.
The sound waves penetrate the body.
The sound waves hit the fibers, they flex, and they generate enough localized voltage and reactive oxygen species to fry the cancer cells wirelessly from the inside out.
The mechanical harvesting applications are endless. You could weave this material into the hull of a deep-sea submarine.
Oh, wow.
And the constant immense pressure of the ocean water squeezing the hull generates continuous silent power for the sub.
Wow.
Or you could coat the skin of hypersonic aerospace vehicles in it.
And generate power from the friction.
Exactly. Generating massive amounts of power directly from the intense aerodynamic friction and vibration of flying at Mach 5.
But coming back to the nuclear battery aspect, I have to jump in here with a massive reality check.
Okay, let's hear it.
Because on paper, this sounds perfect, but we are talking about growing uranium-laced protein batteries in vats of algae.
Yes, essentially.
Because the manifesto explicitly talks about using cyanobacteria in giant open-air bioreactors powered by sunlight to grow this radioactive material at near zero cost.
It does.
That sounds amazing until you realize the regulatory reality. The red tape would literally wrap around the moon.
It'd be substantial.
Substantial. You're combining genetically modified organisms, self-replicating prions, and enriched nuclear material.
A potent mix.
You can't just build an algae farm for that in your backyard. The EPA, the FDA, and the Department of Energy would vaporize you.
That is an entirely valid pushback and a critical lens through which to view this framework.
Okay.
The regulatory dimension of intersecting biosafety, nuclear materials handling, and environmental release of a self-replicating platform is entirely unprecedented in human history.
Yeah, completely.
And to his credit, Enfield acknowledges this extensively in the text. Early-stage, aggressive regulatory engagement isn't just a good idea. It is a strict architectural requirement for this technology to ever see the light of day.
You'd have to invent a whole new branch of government just to monitor it.
However, Enfield makes a compelling geopolitical argument that the overwhelming economic advantage makes its development inevitable.
Regardless of the red tape.
Regardless of the red tape, consider the economics. If you have a manufacturing process that produces advanced, radiation-hardened, multimodal nuclear energy converters, and the only inputs required are atmospheric carbon, sunlight, and trace amounts of naturally occurring uranium, passively filtered from ocean water.
Right.
The marginal cost of production approaches zero.
It's basically free power.
You don't need a multi-billion-dollar semiconductor fab facility. You don't need highly enriched weapons-grade uranium.
Wow.
If one nation or even one well-funded private corporation figures out how to navigate or bypass the regulations to build these bioreactors,
They win.
the strategic and economic advantage is so absolute that all other nations will be forced to adapt or be left behind permanently. It forces the regulatory hand.
It completely rewrites the global balance of power.
Yes, it does.
Okay, but let's say we navigate the nightmare regulations. We have the indestructible structure, we can shape it with precise light, and we have the ultimate solid-state nuclear power source to run it.
The physical infrastructure structure is complete.
But power is entirely useless without control. If we have the structure and the energy, we need a brain capable of managing it.
We need a computer.
We need a computer. But if you try to run a dense computational network with the kind of energy density a coalescent nuclear transducer puts out,
It gets hot.
it is going to get incredibly hot, which requires a complete rethinking of how computers deal with heat.
And that leads us perfectly into the most theoretically complex section of the manifesto, the 8-layer thermodynamic computer.
This part is wild.
To truly understand why this is necessary, we have to look at what Enfield calls the integration fantasy Steelman.
The what?
He purposefully presents the absolute best argument from his imaginary critics and then systematically dismantles it.
Right, the critics' objection to DNA computing.
Yes.
We all know DNA is the ultimate hard drive. Biology figured out data storage billions of years ago.
Very efficiently.
DNA can store insane amounts of data, 2 bits of information per base pair. You could theoretically fit the entire data of the modern internet into a single shoebox full of DNA.
True.
But storage isn't computing. To read and write that data fast enough to act like a modern computer processor, Enfield's framework proposes using terahertz radiation.
Terahertz.
Terahertz pulses are microscopic flashes of energy that can literally unzip and rezip the hydrogen bonds of DNA in picoseconds.
Blindingly fast.
It's incredibly fast.
But the critic points out a massive, inescapable flaw in physics. Terahertz radiation is heavily absorbed by liquid water.
And DNA only functions in an aqueous environment. It has to be wet.
Yes.
If you dry it out, it becomes useless dust.
Exactly. So the critic says if you blast wet DNA with high-frequency terahertz radiation to read and write data, the surrounding water instantly absorbs the radiation.
Which inverts it into heat.
Yes. And classical computing absolutely despises heat. Heat creates thermal noise, it disrupts the precision of the electrons, and eventually it just melts the physical substrate.
Right.
The critic concludes this architecture is impossible. The required heat destroys the classical computing mechanism. It's not a solvable engineering problem, it is a hard limit of physics.
This is where the teardown of that argument is so incredibly satisfying.
It is.
The critic is absolutely correct that it destroys a classical computing architecture.
Yeah.
But this isn't a classical computer. Enfield argues this is a thermodynamic computer. The massive breakthrough here relies on a 2025 Nature paper published by researchers at Caltech, Song, and Keenan.
Right.
They proved that in this specific molecular environment, heat is not the enemy. Heat is the battery.
Let's explore why classical computers hate heat to understand the contrast.
Okay, let's do it.
In a classical silicon computer, we spend massive amounts of energy constantly fighting thermal noise to maintain deterministic binary states.
Right, ones and zeros.
A switch must be a strict 0 or a strict 1. If it gets too hot, the electrons jump around, and a 0 accidentally becomes a 1.
And the system crashes.
System crashes.
Yeah.
This is exactly why giant server farms require massive cooling towers and liquid nitrogen systems.
But in a thermodynamically favored molecular computer, the paradigm is flipped.
Yes.
The system is physically designed so that the correct computational output naturally corresponds to the lowest free energy state of the system.
I believe you had an analogy for this earlier.
Yes. Think of a ball rolling down a bumpy hill into a valley. The correct answer to the math problem is the bottom of the valley. In a normal computer, you have to precisely calculate and force the ball every inch of the way down.
Right.
If the hill starts shaking, which is heat, the ball bounces off course and you get an error.
A fatal error.
But in a thermodynamic computer, the shaking is what helps the ball find the lowest point.
Oh, I see.
The thermal fluctuations, the heat, actually jiggle the system, driving the molecules to snap together into the most comfortable, lowest energy state, which is the correct answer.
The Caltech team literally demonstrated complex enzyme-free DNA logic circuits that are recharged by temperature cycles.
Recharged by heat.
Yes. You heat the fluid up, which forces the DNA strands apart, resetting the circuit from its stable equilibrium state back to a chaotic out-of-equilibrium state.
Ready for the next calculation.
Ready for the next calculation.
Yeah.
So the terahertz pulse provides the sharp picosecond right command, the water absorbs it and generates a local heat pulse, and that exact heat pulse provides the thermodynamic energy to reset the DNA state.
It runs on the thermal noise, not against it.
The heat doesn't melt the computer.
Yeah.
The heat cranks the engine.
It is brilliant. So what does this entire computational stack look like when it's all put together?
It's massive.
Enfield breaks it down into an 8-layer stack. It is so comprehensive it makes my head spin. Let's walk through it, layer by layer, starting from the atomic level.
Layer 1 is quantum electron tunneling. This is the tyrosine hole hopping we discussed earlier.
The stepping stones.
Right. It provides the raw, foundational electrical charge transport across the fibers.
Layer 2 is piezoelectric transduction. This is the material converting the mechanical vibration of nuclear decay or ambient sound into the energy needed to drive the quantum tunneling.
Layer 3 introduces RNA as real-time execution.
Okay, let me use an analogy here to break down the relationship between DNA, RNA, and the prion fibers for you listening.
Go ahead.
Think of your laptop or your smartphone. The DNA is your hard drive. It is the massive, incredibly stable, long-term quaternary storage where all your files live. The T-Hertz laser pulse is the read-write head accessing the hard drive.
Okay.
The RNA, because it is more fragile and temporary, acts as your RAM. It's the dynamic, disposable working memory actively running the apps right now.
Perfect.
And the prion fibers. They are the physical motherboard. They are the tough, conductive wiring, passing the signals between the hard drive and the RAM via physical shape changes.
That mapping is highly accurate to the biological function, which brings us to Layer 4 prion conformational logic.
Prion logic.
The physical cascading shape changes of the amyloid fibers aren't just wires. They can act as physical logic gates.
Like A&D gates and OR gates.
Exactly. Processing information purely through geometry. Layer 5 is the thermodynamic heat recharging we just analyzed, the engine of the system.
The bumpy hill.
Right. Layer 6 is the deep DNA and tiered storage layer.
And layer 7 is photonic quantum programming.
Yes.
This is where the framework loops back to the optogenetics and adds quantum computing. Because the amyloid fibers naturally twist as they grow, they form microscopic helices.
Like a spiral staircase.
Right. And because they are helical, they can act as chiral waveguides.
Explain chiral waveguides for the audience.
Basically, chirality means handedness, like your right hand and your left hand.
Okay.
Light can also have handedness. It can spin to the left or spin to the right as it travels.
Circular polarization.
Exactly. Because the protein fiber is shaped like a spiral staircase, it can physically separate light.
Oh, wow.
It can route left circularly polarized photons down one pathway and right circularly polarized photons down a completely different pathway.
That's incredible.
And being able to cleanly route and entangle individual photons based on their spin is the absolute foundational requirement for building a photonic quantum computer.
All grown out of a vat of biological protein.
It's absurd.
And finally, Layer 8 is civilizational infrastructure. The whole system scaled up to macroscopic size.
But before we scale up to civilizational size, we have to talk about why this 8-layer biological setup is necessary. Why is it so much more efficient than the best silicon chips Apple or NVIDIA can make today? It all comes down to Landauer's principle.
Rolf Landauer was an IBM physicist who established a fundamental law of information theory and thermodynamics in 1961.
Okay.
He proved that erasing a bit of information has an unavoidable minimum thermodynamic cost.
Erasing data costs energy.
It physically desiccates a specific amount of heat. Classical silicon computation is inherently destructive.
That's how.
When a standard logic gate like an AND gate processes two inputs into one output, information about the original inputs is forever erased and lost.
Oh, because it turned two things into one thing. You can't go backwards to find out what the two things are.
Exactly. That erasure generates heat. Billions of microscopic transistors erasing billions of bits, billions of times a second.
Wow.
That is exactly why the processors inside giant AI server farms run hot enough to boil water and require massive cooling infrastructure.
We are constantly paying a physical heat tax for forgetting things. Every time your computer clears its memory, it pays a tax in heat.
Exactly. But the biological architecture Engfield is proposing utilizes what is effectively holographic information geometry.
Holographic geometry.
It preserves the state of the computation via physical geometric conformation.
Okay, break that down.
When a prion fiber cascades and folds the next protein, the upstream protein doesn't lose its shape. It simply passes the shape down the line.
Oh, so it just copies it.
It is a physical copy operation, not an erasure. Because the system preserves the geometry and doesn't erase information, it doesn't pay the Landauer penalty.
It doesn't pay the heat tax.
It approaches the theoretical thermodynamic floor of efficiency. It is computing that is so perfectly efficient, it practically owes you energy back.
It is the ultimate closed-loop system. It builds itself out of light and cheap protein. It powers itself with trace nuclear decay and mechanical vibration.
Yes.
And it thinks for itself at near-perfect thermodynamic efficiency, using its own waste heat as a battery. It's incredible.
It truly is a marvel.
So What does this all mean when you take this perfect microscopic smart node and you launch it into space? Because that is the end game of the manifesto. We are moving into Section 5, scaling to the stars.
This is where the framework transitions from complex material science into the realm of speculative civilizational engineering.
Right.
And to cross that bridge, we must introduce the holographic principle.
A holographic principle.
In advanced physics, derived from Bekenstein and Stephen Hawking's work on black hole thermodynamics.
Okay.
The holographic principle posits that the complete information of a three-dimensional volume of space is entirely encoded on its two-dimensional boundary.
The part intrinsically contains the whole.
Exactly.
As a physical glass hologram.
Yeah.
If you have a holographic plate with a 3D image of an apple on it and you drop it, it shatters into 100 pieces.
Right.
But if you pick up one tiny shard and shine a laser through it, you don't see a piece of an apple. You still see the entire complete apple, just at a slightly lower resolution.
That's a great
And
example.
every piece contains the whole blueprint.
Precisely. Enfield argues that the cross-beta amyloid scaffold is a physical, biological manifestation of this exact principle.
Oh, so?
The tip of a single microscopic prion fiber, chemically and geometrically, encodes the entire future architectural blueprint of the massive macroscopic structure it will eventually become.
Whoa.
The geometry of the tip is the instruction set. If you drop one microscopic seed fragment onto a substrate, it contains all the information necessary to grow the macrostructure.
And this concept completely changes how we measure the advancement of a civilization.
It really does.
If you're a sci-fi fan, you've probably heard of the Kardashev scale.
Sure.
It's how astrophysicists rank alien civilizations. A Type 1 civilization harnesses all the energy of its home planet.
Right.
A Type 2 harnesses all the energy of its home star. A Type 3 harnesses the energy of its entire galaxy. It's an entire metric based purely on aggregate energy consumption.
But Enfield proposes a profound recalibration. He defines what he calls a biological Type III node.
A Type 3 node.
He argues that a truly galactic civilization isn't defined by how much raw wattage it consumes.
Okay, what is it defined by?
It is defined by its supply chain. It is defined by whether its fundamental unit of the node requires a planet-bound factory to reproduce itself.
Think about human civilization right now. We cannot build a galactic empire today.
Not even close.
Why? Because if we want to build a single spaceship, we need lithium mines in Africa. We need highly sterile silicon fab plants in Taiwan.
We need massive steel mills in China.
Right. And we need a multi-billion-dollar launch pad in Florida. The supply chain is inescapably bound to the gravity well of the Earth.
But if your entire technological stack manufacturing, computing, power generation, structural materials is encoded in the DNA of a biological organism that can build piezoelectric amyloid infrastructure using nothing but basic atmospheric carbon, ambient nitrogen, and starlight.
You don't need a supply chain.
You don't need a supply chain. You just need a single seed.
Which brings us to the ultimate application, the piezoelectric Dyson swarm.
The Dyson swarm.
The classic concept of a Dyson swarm is a massive cloud of billions of solar panels surrounding a star to catch all of its energy output.
Yes.
The problem has always been materials. Physicists calculate you would have to dismantle entire planets like Mercury and Venus just to get enough raw metal to build the panels.
It's structurally and economically impossible.
Unless the panels grow themselves.
Right. You don't build a massive fleet of construction ships. You take a tiny, compact seed package of this engineered biology, roughly the size of grapefruit.
The grapefruit.
And you shoot it at a carbon-rich asteroid in a distant solar system. It lands. The biology activates.
And it just starts eating.
It begins eating the carbon and nitrogen in the asteroid. It uses the starlight for photosynthesis, and it begins growing massive, delicate, piezoelectric amyloid collector membranes in the vacuum of space.
Wow.
Because it's biology, it replicates exponentially. One panel becomes 2, 2 panels become 4, 4 become 8.
Then 1,000, then 1,000,000,000.
Exactly. It entirely consumes the asteroid, grows into a massive solar collector swarm, and powers itself.
And because the material is intrinsically piezoelectric and chiral, it isn't just a passive mirror harvesting photons.
Yeah.
It is harvesting the mechanical pressure of the solar wind, hitting the sails. It harvests the thermal expansion and contraction as it orbits.
The thermal gradients.
It harvests the gravitational tidal forces of the star pulling on the massive web. And because those fibers act as electromagnetic waveguides, the swarm isn't just collecting energy.
It becomes a coordinated, stellar-scale electromagnetic field projector.
This leads to stellar engineering. You aren't just passively sitting around a star collecting a suntan, you are actively managing the star.
Yes.
The framework talks about using the swarm's massive coherent electromagnetic fields to essentially squeeze the star.
A managed fusion reactor.
It's like a stellar tokamak. You use acoustic resonance, literally using magnetic fields to push on the star's natural internal sound frequencies.
To increase the density of the plasma core.
You can speed up or slow down the fusion rate. You literally have your hand on the throttle of a sun.
And the communications infrastructure is equally staggering. They invoke the Gertzenstein mechanism.
Okay, the Gertzenstein mechanism. Explain how this works, because this sounded like absolute magic when I read it.
It is not magic. It is a validated, though highly advanced, physics principle formulated by Mikhail Gertzenstein in 1962.
Okay, what does it do?
He demonstrated mathematically that in the presence of a massive static magnetic field, high-frequency electromagnetic waves can actually be converted into gravitational waves.
Wait, really?
Yes, a Dyson swarm made of highly conductive chiral biological fibers could generate a magnetic field the size of a solar system.
Wow.
By pulsing electromagnetic signals through that field, the swarm could theoretically convert its radio transmissions directly into ripples in the fabric of space-time, gravitational waves.
But why would you go through all that trouble? Why not just use lasers or radio waves to talk to your other star systems?
Because the galaxy is incredibly messy. Radio waves and lasers are easily blocked, scattered, or absorbed by interstellar dust clouds, rogue planets, and the glaring radiation of other stars.
Oh, that makes sense.
But gravitational waves are not blocked by anything. They pass cleanly through dust, gas, stars, and even black holes.
Nothing stops them.
It provides an unblockable, permanent, galactic-scale communication channel. It is the internet backbone for a Type III civilization.
Okay, I have to stop you. I have to pull the emergency brake right here.
Oh, heck?
Because everything we are talking about sounds like we are building a godlike machine.
It certainly sounds like it.
We are shooting biological seeds at asteroids that exponentially consume matter to build galaxy-spanning computers that manipulate stars and talk through gravity.
Yes.
How do we stop it from pulling a gray goose scenario on a cosmic scale? What if the seed misses the asteroid and lands on an inhabited planet?
Catastrophic failure.
What if it just consumes the Earth and all the trees and all the people to make more solar panels?
It is the ultimate existential risk question. And the synthesis requires us to revisit the kill switch architecture we briefly touched on earlier.
The caps and plugs.
Yes, but scaled up to a planetary defense level. A civilization capable of engineering this would absolutely require self-limiting geography built into the seed at the deepest genetic level.
What does that look like in space?
We are talking about highly redundant CRISPR-mediated dissolution agents and genetic dominant negative inhibitors programmed to respond to environmental triggers.
Okay.
The biology must be programmed to only activate in a hard vacuum or on very specific asteroid chemistries.
So it won't grow in air.
And more importantly, it must be programmed to trigger its own complete structural breakdown, literally dissolving back into inert amino acids.
Yeah.
If it detects atmospheric signatures, liquid water oceans, or specific biochemical markers of natural life.
It has to have a self-destruct.
It must be designed as a closed, tightly bounded thermodynamic loop.
It has to have the brakes built deeply into the DNA before you even think about installing the engine.
Precisely. If we connect this back to the core philosophy of the entire framework, this isn't just an infrastructure project. It fundamentally dissolves the boundary between the builder and the built.
How so?
Throughout human history, we are used to humans being the operators and the buildings or the machines being the static objects.
Right.
But here, the living organism is the infrastructure. By encoding the entire civilizational tech stack in the biology of a single microscopic node, the civilization is conceptually Type III the moment the biology works in a lab.
Wow.
Because once that single seed works, scaling it to conquer a galaxy is no longer an engineering problem. It is just a matter of basic arithmetic and time.
Wow. Okay, let's take a breath and bring this massive galactic scale back down to the human level.
Probably a good idea.
Let's recap what we've unpacked today. We started with a simple misfolded protein, the exact kind of glitch that causes devastating brain diseases. We learned how to isolate its structural superpower to build Kevlar-like nanowires. We learned how to shape and control its growth using lasers and optogenetics. Right.
We turned it into a solid-state nuclear battery. We turned it into a thermodynamic computer that runs on heat and approaches perfect efficiency.
Yes.
And finally, we saw how that single biological seed could be launched into space to exponentially build a Dyson swarm and manipulate the fusion rate of stars.
It is a profound, almost dizzying demonstration of how deeply understanding the physical geometry of information at the nanoscale can unlock engineering capabilities at the astronomical scale.
Yeah,
It shows how the micro dictates the macro.
it really does. And I want to leave you, listening to this right now, with a final provocative thought.
Okay.
If Enfield's total application framework actually comes to pass, if we truly integrate human brain-computer interfaces with this biological tech stack in the future, you wouldn't just be using this technology like you use a smartphone. Through neural interfaces and medical nanites, your own cells, your own body, would become part of the conducted infrastructure.
It'd be part of the network. You would literally carry the entire blueprint of a galactic civilization inside your biology. So the question to mull over tonight is this. In a world where the living organism and the technological infrastructure are completely identical, does the civilization exist to serve you, or are you just a walking, breathing node existing purely to serve the civilization?
A haunting and entirely necessary question for the future.
Thank you so much for taking this deep dive with us. We'll see you next time.